I've only got 70 slides, so we should be able to get through them. So distinguished chairs and organizers, thank you very much for the invitation to come here. I've been hearing good things about ICRISAT for my entire professional career, and it's a real pleasure to be here. So, with that, since the maize reference genome was published in 2009, the amount of resequencing data has exploded. So this is up through February of last year, so about a year ago. We had already about 10 terabases of maize resequencing data available in public databases. Now, of course, it's more. So one of the key questions in my research group is how to translate genomic data to biological understanding and crop improvement. And that's what I would like to focus on. So there are a lot of ways to tackle this. The approach that we're quite interested in is trying to associate genes with traits, determine which genes control traits. And there are multiple ways to do this, and we're doing a variety of methods, including mutagenesis. But what I'm going to talk about today is using GWAS to try to define which of the 50,000 genes are controlling particular traits. Now, as we have had introductions to GWAS already today, there are a number of different algorithms available for conducting GWAS, and we were interested in trying to understand the strengths and weaknesses of these algorithms, not from a mathematical perspective, but from an empirical perspective. So if we used the same data set and analyzed it with multiple algorithms, which algorithms were giving us the best perspective of truth? So for this project, my former graduate student, Jinling Yang, who's now a postdoc in Jeff Rossavera's lab at UC Davis, selected a number of yield-related traits that affected the maize cob length, weight, diameter, the kernel row number trait, which I'll talk about a little bit more extensively today, and then a number of other traits such as kernel count, total kernel weight, and the average kernel weight. So we used the NAM population developed by Ed Buckler and his colleagues. I won't go through the development of that population. We've heard already about NAM and NAM-style populations here at the conference. But in addition to the 5,000 NAM RILs, we also backcrossed a number of the RILs to Missouri 17 and also to B73, the reference line that was used in the construction of the NAM set. And then there was a key population we added which allowed us to do something quite interesting that I will come back to, which is that we made a partial diallel of the founders, the 27 founders of NAM. We used these to create a partial diallel. So we'll come back to that. One of the beautiful things about working with this population, in addition to its powerful genetics, are the genomic resources that have been developed for this population. So Ed Buckler and his colleagues have already generated millions of SNPs for this population. We went into the founders and did deep RNA sequencing on multiple tissues from each of the founders and generated about 4.9 million SNPs from each of these founders. And so when we put all of the SNPs together from the different data sets, did some merging and filtering, we ended up with about 13 million SNPs that could be projected onto all of the lines, both the RILs themselves as well as the backcrosses and the partial diallel lines. So genotyping was taken care of. We also needed trait data. And for this, we downloaded some data from Pat Brown's PLOS Genetics 2011 paper. He had collected some of these phenotypes on the NAMs per se. We also generated a large amount of phenotypic data ourselves for these particular traits, ultimately ending up with about 100,000 observations from 9 locations. And consistent with what we're hearing at this conference, the genotyping was much easier than the phenotyping. And I'll come back to that a little bit later in the talk. Once we had the genotyping data and the phenotyping data, it was possible to do GWAS using these 3 algorithms. And I'm not going to go through the details of how we did that, or even to any large extent the results of that. But I do want to talk about cross-validation. So we do GWAS experiments and we always wonder, are we discovering truth or are we getting false positives? So what we did is we took a subset of the SNPs that came out of these GWAS experiments, and we looked in independent populations, unrelated to NAM, and asked whether we would find associations with these SNPs with the traits of interest. And so about half of the SNPs that we tested in this way could be confirmed. Now this would be a minimum estimate of truth. So at least 50% of the SNPs that came out of these GWAS experiments could be cross-validated in independent populations. So this, I think, was very good news. We also compared the level of truth that each of the three algorithms produced. So this plot is a little complicated, but on this axis here is the single variant GWAS results, and anything to the right of this dotted line was called significant according to this algorithm. And so if we wanted to know how well this algorithm did, we would ask for all of these spots, how many of them could be cross-validated. And if we look on this axis, everything above this line was called correct. There was a true association. There was an association called by the Bayesian method. And we have classified blue are those that were genotyped but not cross-validated, and the red triangles are those that were cross-validated. So the Bayesian approach, about a third of the SNPs that we called with that method that were not called by the single SNP method could be cross-validated. The single variant, about 2/3 of them, though the numbers are quite small, could be cross-validated. And if both of these methods called, we had about a 75% chance of being cross-validated. We also looked at the stepwise regression. Now the key take-home here is not these percentages, because I don't have a great deal of faith in those because the numbers are small, so there are large error bars associated with these. The key thing is that each of these algorithms is finding a different perspective of truth. So it's like the blind guys and the elephant. You know, each method is finding SNPs associated with the traits that the other methods did not find, and that these are true. These are true cross-validated SNPs discovered by one method and missed by other methods. So this is an important finding. Ultimately, we had run the GWAS for all 7 traits, and we ended up with over 1,000 associations scattered across the genome. Now, of course, these associations are useful for biological understanding of the traits. They can also be used as markers for breeding programs. But really what I want to talk about today is not these associations per se, but the use of those to gain some understanding in heterosis or hybrid vigor. So this is something as a community we've all been trying to understand for well over 100 years. Why is it that certain inbreds, when crossed together, produce heterotic hybrids? It's certainly not true that any pair of inbreds gives us a good hybrid. One of the ways that the big seed companies make their money is by figuring out which pairs of inbreds should be crossed to produce hybrids. And to a large extent, it's still a trial and error process. Large numbers of inbreds are crossed and yield tested to identify the really superior pairs. And this uses up a great deal of breeding resources, is this trial and error approach. So what we'd like to do is understand the fundamental biology behind underpinning heterosis, just as Doug Cook mentioned earlier before the lunch break. This basic understanding can guide our applications. But more immediately, we'd like to understand heterosis to the level where we can predict hybrid performance. So we can identify pairs of inbreds and predict with some accuracy the performance of those hybrids, which are going to be the winners and the losers. I do not have an illusion that we will be able to pick the absolute winners with this approach. But if we can simply identify the bad 50%, the bottom half, and not even waste our time field testing those, we will have made a contribution. So, the amount of heterosis that we see for these traits varies. So Cobb diameter and kernel row number exhibit essentially no heterosis, where total kernel weight has substantial amounts of heterosis. So this has been known for a long time. Different traits exhibit different amounts of heterosis, and of course we see different amounts of heterosis in different crosses, as I'd already said. So we have SNPs, and we already know the phenotypes. We can ask, how well do the genotypes predict the phenotypes that we already know? And that's what's plotted over here on the left. So each of the traits is plotted here, and on the y-axis is how well the genotype can predict the phenotype. And it ranges from about 0.7 down to about below 0.4. So it's correct that the genotype is helping us predict the phenotype, but it's not very good at this stage. But we also notice something interesting, which is you'll see that these are plotted along the x-axis. We are plotting each of these traits based on the percentage of high parent heterosis. And what we can see is as the percentage of high parent heterosis for a trait increases, the ability to predict the performance of that trait declines. So this is essentially a missing heritability. We can't predict this phenotype. And our ability to predict decreases with traits that have high heterosis. Now that was using all 4 of the GWAS populations that we had. If we focus now just on that partial diallel population where we have all 3 possible genotypes, small a, heterozygous, large A, large A. In a diallel we get all 3 genotypes. And we know these genotypes because we have all of these SNPs that we can project onto the hybrids. What we can do here is we can add into our prediction not simply an additive genetic model, but we can add dominance into our prediction. And if we do that, this curve's a little different from here because we're only using 1 population instead of 4 here. But the general pattern is the same. If we have only additive gene action in our model, we do better at predicting the low heterosis traits than the high heterosis traits, but we don't do terribly well with any of them. If we add dominance in and combine these 2, we find that we actually do very well. Now we're in the sort of 0.9 to 0.95 or 0.85 to 0.9 accuracy of predicting our phenotype based on genotype if we include dominance in our model. Now this is not surprising given the classical models for heterosis, which often implicated dominant or overdominant gene action. But I wanted to go a little further than this, and we wanted to look at the effect of individual SNPs and their gene action. So for each one of the SNPs that was based on our GWAS, each individual SNP that was associated with a trait, we could classify the gene action of that SNP as being strictly additive. So if we have the 3 genotypes in the partial diallel population, if the heterozygote is absolutely at the midpoint of the 2 alternate homozygotes, this SNP, this particular gene, would have truly additive gene action. If, on the other hand, the heterozygote was superior to the average of the 2 homozygotes, we would have positive dominance. And if the heterozygote was inferior to the average of the 2 homozygotes, we would have negative dominance. So every one of the SNPs was classified in this manner. Now, this is the most complicated slide today, and if you're taking a little rest after lunch, now's the time to wake up for just a few minutes. All right, so here we have all of the traits, and we have organized them based on the amount of heterosis that they exhibit. So at this end, kernel row number and Cobb diameter have essentially no hybrid vigor, no heterosis. And total kernel weight has large amounts of heterosis. For each of the genes, each of the traits, we have identified a number of SNPs that are associated with that. So for example, for kernel row number, we have 189 SNPs that we associated from our GWAS experiments as contributing to this trait. Of those 189 SNPs that associate with kernel row number, 58 of them show positive dominance. 79 of them show negative dominance, and 52 of them show additive gene action. On the other extreme over here, where we have total kernel weight, which has a large amount of heterosis, we had 127 SNPs associated with this trait, and 99 of those, or 78%, show positive dominance. Only 5% of them show negative dominance, and 17% show additivity. So there's a pattern here, and we can see this with the pie chart here, that the proportion of green, which are the additive SNPs, the proportion of SNPs contributing to a trait that are additive is approximately equal across the categories. But the ratio of red to blue, red being positive dominance and blue being negative dominance, changes dramatically as we move across the spectrum and move from low heterosis traits to high heterosis traits. So what this is telling us is that the traits that show high heterosis have a preponderance of loci that are showing that the ratio of additive to dominant doesn't change. But of the dominants that contribute to total kernel weight, most of them give positive dominance and very few give negative dominance. Where low heterosis traits, the ratio is approximately 1:1. So we believe that this is a step towards helping us to do a better job predicting which inbreds, when crossed together, will give us high-performing hybrids. Because we don't now look at every SNP in the genome, we can focus in on those SNPs that are known to affect particular traits, and we can understand the gene action associated with those particular loci that affect the particular traits, and we can identify parents that when crossed together will give us heterozygous genotypes that will give us the positive dominance. Now, of course, predicting phenotypes of hybrids is not simply a matter of genotype. We also need to concern ourselves with environment, which people have been doing for a long time, but also genotype by environment interactions, which makes this much more difficult. So there are a number of different strategies for dealing with the effects of environment and genotype by environment interactions on predicting phenotypes. And one of the strategies, which we've taken advantage of when we look at kernel row number, is simply to study traits that are stable across environments. Kernel row number is essentially not affected by environment at all. So if we want to study that trait, we don't have to worry about the environment. It's just not an issue. Another strategy that some people take is to do their experiments in growth chambers or other controlled environments where they simply take environment out of the equation. They do their entire study in a single environment, which removes both the E and the G by E terms, and then we can just look at the effect of genotype on phenotype. And this is certainly an important way of approaching these studies. But we've taken a different approach. Nils Stein will recognize perhaps this view of the IPK, and I was absolutely amazed when I visited the number of greenhouses they have. In Ames, Iowa, where I am, we don't have very many greenhouses, but we have a lot of land. So we're proposing to do these experiments not in controlled environments, but to do them out in the world, the real world, and control for and study the effects of both environment and G by E statistically to gain a deeper understanding of how these affect plant growth and development. So to do that, we need field-based phenotyping platforms. I mentioned that for the NAM population and the GWAS, generating the phenotypic data was much more complicated than generating the genotyping data, and this is because we were doing it almost by hand, almost manually. So we've been moving towards automated or higher throughput systems, and I'm going to talk about a couple of these very briefly here. So the first is to mount sensors on field-deployed robots, which is relatively expensive, but it has some interesting advantages. So this is a project that I'm conducting with my collaborators, Lee Tang, who's an agricultural engineer, and Maria Salas, who is a sorghum breeder at Iowa State. And we've taken what is a commercial-grade lawn tractor, and it looks like a tractor here, but it's not. It's really a robot. And the reason it's a robot is it has a GPS unit here, and it has an auto steer unit attached. So we can program this thing to drive through the field autonomously. On the front here, we have a rack upon which we can put a wide variety of sensors, for example, time of flight, and some others that I'll show you in the next slide. So we can program this robot to drive through the field collecting data from our plants. So for example, we've put near-infrared stereo cameras. This is actually 2 cameras here, and we can get a 3D image in the near-infrared wavelength of the plants, which just allows us to very easily distinguish the plants from the ground. All right, now, there we go. I want to show you what this looks like. There. So we call this the Phenobot, and here is the robot moving through the field. There is this rack here, and the sensors are on the rack, and you can see that it's driving all by itself. So we don't need any staff anymore. We just drive this thing through the field, and if we can get it to send the data back to the lab, we'll never even go to the field anymore. Okay, so that's one approach, and it has some particular advantages, but it is relatively expensive. So we're also looking at some inexpensive field-based sensors. And this is being done in collaboration with James Schnable, who's a professor at the University of Nebraska, and my postdoc, Young-Suk Chung, at Iowa State. And this was really James's brainchild. So he was working with these Raspberry Pi computers. They cost something on the order of $30 a piece. You can put a cell phone camera on this. You can also put an SD card, so you can program. This is a fully programmable computer. You can program it to take a picture every 5 minutes and then timestamp the photographs and store them on the SD card. And then you can put those together to create stop-action movies. Now, this is a collaboration, so both parties have to contribute, right? So he built all of this. I built this nice wooden adapter here that goes onto the tripod. Now, this is a very early version. My postdoc has developed much more advanced technologies. We did our first field experiment this winter in Hawaii. We do a winter nursery in Hawaii every year. And so the first pilot was done in Hawaii. Young did them this year. This summer we plan to deploy 300 or 400 of these sensors in the field so that we can measure phenotypes. But I want to show you some data that James collected in the greenhouse. And this is a dynamic response to drought. There we go. All right, so these are plants in the greenhouse, and they're experiencing drought. James stopped watering them. And what I'd like you to do is focus on the lower leaves here. A photograph is being taken every 5 or 10 minutes, and they've been stitched together to create this video. And let's just keep watching these lower leaves here. Look what's happening here. Look at this. Now, I've been looking at corn plants, and I've seen the effect of drought for decades, longer than some of you have been doing science, probably. And I've never seen this before. What this allows us to do, instead of looking at the endpoints, the endpoint phenotypes, we can watch the process of phenotypes developing. We're compressing real time into a time that allows us to observe the phenotype developing. Now, one of these plants is very different than the others, and it actually has a transgene in it, which I don't care anything about, but it was in there as a control. The point is, this gives us a dynamic response. We can watch plants develop, so we could watch, for example, flowering take place. We can watch plants respond to their environment and begin to do, for example now, GWAS to understand not the endpoint of a trait, but how it progresses. All right, and then I want to just mention very quickly unmanned aerial vehicles, or drones as they're called sometimes. And I don't have data here. There are really complicated regulations to use drones for science in the United States right now. But this is a drone that I bought at Christmas for my son. It was about $1,000 from Amazon. And in 5 minutes on Christmas Day, we went outside, we launched this. There's a high-D HD camera attached to this, and we were collecting data about our neighborhood 5 minutes after we opened the box and charged the thing up. Now, I need to go through the regulations to be able to do this for real in an experiment, but right now I can go flying around the neighborhood with no regulation. Okay, so where does this leave us? If we're able to develop these predictive models using genotype, environment, and G by E, we'll be able to improve the accuracy of selection in plant breeding experiments, which should increase the rate of genetic gain per year. It should enhance our ability to breed crops to withstand climate change, and we should be able to give farmers better evidence-based recommendations for the appropriate varieties or hybrids to grow in a particular field under a particular management practice in a given year, which should lead greater farmer profits and, importantly, enhanced yield stability. So to summarize, DNA sequence variation can explain 40 to 70% of genetic variation if we only consider the additive gene action, or 80 to 90% if we include dominant gene action in our models. These dominant effects explain much of the missing heritability that we observe if we only use additive gene effects. The ratio of loci exhibiting positive dominant gene action to those loci exhibiting not negative dominant gene action is correlated with the degree of heterosis for that trait. So determining which loci have positive and negative heterosis for specific traits may increase our ability to predict hybrid performance. Phenomics is clearly, as we've heard from lots of people during the conference, phenomics is a bottleneck for GWAS, genomic selection, and breeding in general. We believe these field-based sensors will allow us to study the genetics of dynamic traits rather than being limited to endpoint traits. And then I just want to acknowledge this is my team at Iowa State University. I also have a lab at China Agricultural University, and these are those guys. I'd be happy to answer questions.
Questions? So, where?
There you go.
Looked into the variability among the parents as single point or SNPs, basically. Have you also started looking into the variability or the polymorphism as SNP haplotypes and then trying to correlate with whatever you're doing?
So the question has to do with what I've described so far as just looking at single SNPs, but have we looked at whole haplotypes? And we have some very interesting results doing GWAS not based on just single SNPs, but essentially haplotypes, that we're finding a bigger, more interesting picture than just the individual SNPs.
Is it following the same trend which you have got with single SNPs, or is it different?
We haven't gone to the point of validating those and certainly not looking at their effect on heterosis. I'll also say we've got a very clear, detailed picture of presence-absence variation in these lines, and we're also looking at those. Our preliminary findings are that not all of the structural variation is in LD with SNPs, which means that if we only rely on SNPs, we're going to be missing some of the control. Maybe Doug here?
So,
yeah,
I've got the microphone, so I'll go. So, nice talk. So, given that the HPH SNPs are largely positive acting, they're enriched for positive acting SNPs, then you would expect that these things are themselves under selection. So I have two questions. Have you modeled selection across those genes versus a random set of genes in the genome? And then the second one is, if you go into wild populations, I'm wondering how many of these SNPs are ancestral, and therefore what might be their role as opposed to derived and functional in cultivated systems?
These are extremely good questions, and this is really why Jinling went to Jeff Rosavera's lab, is because he did some exploration of this. We didn't find strong signals, but the amount of data we had was relatively limited, but it got him excited about those questions, and that's why he's working with Jeff.
So, do you find any correlation between the position of the SNPs on the genome to their contribution to the heterosis?
We have looked, so the question is whether or not the position of SNPs in the genome is correlated with contributions to heterosis. We have looked, it's actually a fairly hard question to model, but we don't see a strong signal on a genetic map. On a physical map, there are definitely interesting things going on because of low recombination. Yeah.
Traits exhibit more positive dominance than the other traits?
I have no idea. We have, the question is why some traits have more positive dominance than others. We have looked at the genes that surround these SNPs, and many of them make sense. So the one we've done this most extensively was kernel row number, because there's a lot of biology known about the genes that contribute to that. It's a meristem and division. And those make sense. Why some give positive and some give more negative, I don't know. And I think that's going to be quite a while before we understand that. I think using this as a tool for prediction is going to come a lot faster. Yes?
From all the previous studies, what we have seen, data from Dovik and others,
Yep.
If we partition heterosis, much of the gains have come from the improvement of the inbuilt lines. Now,
Can you say again? Most of the?
gains have come from the improvement of the
Absolutely. You're absolutely right about that.
Okay. The second part, which you are stressing, is there are two parts of dominance. One is what we call reinforcing dominance. The other is opposing dominance.
Right.
So the question comes here is how we can reduce the opposing dominance so that the positive dominance effects can be increased.
Right. And so the question is how do we select for the positive dominance and avoid the negative? And I think once we know which genes are giving positive and negative, and we know the genotypes of our lines, we can make strategic decisions about which pairs of lines should be crossed together to maximize the number of loci that are going to be heterozygous for positive dominant loci and minimize the number of heterozygotes for those loci that give negative dominance. So I think we will be able to do this in a more strategic way than we've done in the past. And you're absolutely right that the amount of heterosis has not increased over time, that the yield increases in maize are the per se yields of hybrids, of the inbred, excuse me. But the hybrid vigor is still where the profit comes from.
So we have one more quick question. Last.
Pat, there's
David.
there's a lot of variation in maize, and this is being exploited in wonderful ways to be able to develop these improved varieties. However, that variation is finite. Thinking ahead as to what you could possibly do in the future, would it be possible at some point to predict specific modifications you might want to make to the genome which are not currently represented in the germplasm, which could then advance the phenotypes further? I don't know how you would go about doing that, but to actually take a more radical approach and rather than relying on the diversity that's there, use prediction models to say, okay, where is there a lack of diversity and where could diversity be enhanced and what effect could that have on yield?
Yes, so the question is, ultimately we will perhaps, and it seems reasonable, exhaust the genetic diversity of Zea, and can we generate new diversity that we can exploit? And I think the idea that some loci are giving us positive dominance, if we understand how that works and others that give negative, maybe we can begin to design alleles that will give us one versus the other. And I've stopped predicting that things won't work. When PCR was first invented, I bought a machine and PCR, it didn't work. And I said, this technology sucks. It's never going to work. And we had a bad machine. So these things will all happen, I think. All right. Thank you very much.